Welcome back. Now, I would again like to call Dr. Devendra Sehgal to chair the session. And now we have Dr. Dipyaman Ganguly joining us from Kolkata. He is principal scientist at CSIR-Indian Institute of Chemical Biology, Kolkata. He is joining us online, so please welcome him, even though he's not here, with a huge round of applause.
Okay, while we are connecting, Dr. Ganguly is a principal scientist at IISCB Calcutta, and his uh, research area has been in the area of cancer and inflammatory disorders. Fair amount of work in his lab centers around dendritic cells and inflammatory cytokine milieu. He has there are multiple interests in the lab, which includes right from developing small molecules to for therapeutic use.
Hello, everyone.
Uh,
Um, Hello, everyone. I really thank the organizers, Professor Doloi and other organizers, for inviting me here. I'm really sorry for weird reason I couldn't attend this meeting in like physically. I would have loved to do that. Without wasting much time, let me share my screen. I hope all of you can hear me. Uh,
yeah,
Can someone just
we are good.
give me in the chat?
We can hear
Okay.
you.
Thank you so much. Thank you so much. Let me share my screen and okay. Very good evening to the workshop participants as well as other faculties. I know dignitaries are there. Please accept my regards. So we'll talk about bridging the innate and adaptive immune functions. And I know you have been learning about, you just learned about cytokines and antibodies, etc. But I'll take a step back and I'll try to impress upon you in a very briefly and to a great extent, very simply, on how we came to know about these bridging functions and how important they are for driving human immune response in different clinical contexts. So all of us know that whenever we are talking about immunology and immune system, the large question that we are considering is me or not me, right? So that's how we have been introduced to immunology and immune systems function from the high school days. That immune system is essentially making a sense of my boundary and finding out what are foreigns and they are reacting against whenever any foreign entity invades our body. Now, as you know already, so immunology is a very unique subject that it started contributing to human health much before it actually became a subject on its own, right? So if you think of Edward Jenner, the vaccines, they came much earlier before we actually knew a whole lot about immune system, and the immunology as a subject came into being. And one of the major personalities or immunologists who played a big role in making this domain the autonomous subject and asking the pertinent questions about how immune functions work in the body were these two people, Peter Medower from England and Frank McFarlane Barnett from Australia. And in the mid 1950s, they were doing experiments mostly on transplants, and they were trying to do xenotransplants on mice and on other animals. And largely from those data, they came up with the first model of how immune function works or immune system works. And that is called self-non-self model. What was their model? So they found that when they were transplanting, say, skin from one mouse to another, and if they exposed the recipient mice when they're very young age with some cells of the donors, and then came back with the transplant later on in the life, that transplant was accepted. And that led to this idea, which was largely articulated by Medower, that whatever is self or whatever is part of myself or me is determined early in life. And that's how the immune system knows what is part of me and what is not. And based on mostly that logic, immune cells work and they try to get rid of other foreign invasions, bacteria, pathogens, microorganisms, etc. Now, this is the model that we have been learning about all our life, and is actually a very working model. It works in a lot of contexts, and but interestingly, it also doesn't actually work in a lot more clinical context like cancer or autoimmunity or even pregnancy, right? So and there were a need for a standard model. We'll not discuss into that, but because these questions were there and people really were only thinking about the lymphocytes, the dense nuclei containing cells that are circulating in the blood and present in lymph nodes as the major immune cells. And that was more of a lymphocyte-centric world at that time. And they also figured out that this self-non-self discrimination can be instituted by these lymphocytes, and these are the cells that are going through the central tolerance mechanisms early in life. And so the question was, if this is the model, then why this kind of clinical context like autoimmunity will happen? Because anyway, the self proteins have been recognized early in life. And the first person who changed this model was Melvin Kohn, and he mostly theoretically proposed that this problem can be circumvented if there are other lymphocytes which have largely licensing function. Now, so he introduced this licensing function lymphocytes, and now we know that he was actually talking about follicular helper T cells, and which was, I mean, it was 1969 when he proposed it, and that was the first modification to the self-non-self model, and but these cells were actually discovered much later, 2006, and in fact, when I was a PhD student, then helper T cells were discovered. So that tells you the genius of Melvin Kohn, who actually thought of this licensing lymphocytes and how they can actually mitigate some of the questions that the self-non-self model was facing. Nevertheless, it actually needed more modifications. I mean, the idea about only lymphocytes having to do with self-non-self recognition was not very well supported by the experiments that were being done in 1970s. And in 1970s, MHC class 2 molecules and HLA molecules were discovered. They got Nobel Prize, Zinkernagel and Doherty. And also there were labs which were trying to do the similar transplantation work and trying to find out how these lymphocytes are reacting against different xenotransplants. Interestingly, Alistair Cunningham and Kevin Lafferty, they worked in Australia. They found a very weird finding in their experiments. They found that the T cells respond more strongly against foreign cells of their own species, rather than against cells from another species. And that was really counterintuitive, right? So because if two species are different, they are more distant, their antigens will be more distant, and then the lymphocytes should actually react similarly, if not more vigorously, against another species. But usually does not happen for lymphocytes, and that led them to propose that a species-specific proliferation signal is passing between the cells. That means the cells are not only, these T cells or T helper cells are not only licensing these B cells or antibody producing cells or other effector cells, but they are also perhaps giving a species-specific signal. And they also propose that this species-specific signal is perhaps coming from another cell, which they largely named as, that was largely known as accessory cells at that point of time. And they proposed the term co-stimulation. And so these accessory cells, so the T helper cells are stimulating or licensing the lymphocytes that are effectors. And the antigen present or the accessory cells are producing co-stimulation or other species-specific signals, which are actually licensing both these lymphocyte subsets. Now, that was a new cell that came in, and here you are actually trying to, I mean, you're actually looking at a cell where there is no antigen specific receptor. I know you will learn about antigen specific receptors in a later class, but now we know these accessory cells are antigen presenting cells. Now these antigen presenting cells do not have antigen specific receptors, which are defined early in life, etc. And that creates another problem, because if they are not specific to antigen or any bacteria or in a virus, how will they license the whole immune response? And that was so Cunningham and Lafferty brought about this second modification in the self-non-self model, where they introduced the idea of co-stimulation provided by accessory cells. Now the question was, what actually drives these accessory cells to provide this co-stimulation? And that was largely an unanswered question for a long time, until the gentleman on the left, Charles Janeway, again contemplated that there may be, they may not have antigen specific receptors, these accessory cells, but they may have receptors which can determine if they're seeing an organism or an entity which is evolutionarily distant. And he proposed that there will be patterns on evolutionarily distant organisms like bacteria, viruses, etc., parasites, fungi, and those patterns, which he termed pathogen-associated molecular patterns or PAMPs, will be recognized by these receptors on these accessory cells, which are not antigen-specific, but they can recognize PAMPs. And he termed it as pattern recognition receptors. And the first of the pattern recognition receptors that was discovered was by his postdoc, Ruslan Medzikov, in his lab. That was TLR4. In parallel, another lab, Bruce Bartler's lab in California, also cloned this, one of the first PRRs, which is again TLR4. And so there was a big change in the model. So now we are not concerned about whether the immune system is looking at self entities or non-self entities. Rather, the immune system is looking at evolutionarily distant entities that can cause disease, usually the pathogens. And naturally, this model, Janeway was terming as infectious non-self model, not self non-self model. And that was a big success in terms of explaining a whole lot of clinical conundrums. Now, within 20 years or so, we now know that there are so many pattern recognition receptors that have been discovered. And a whole lot of families are there: toll-like receptor family, Regai-like receptor family, which is mostly cytosolic, node-like receptors, C-type lectin family receptors, which are again on the surface of the cell. And so these are Bruce Bartler. Among them, they discovered a whole lot of pattern recognition receptors. I hope I'm not getting any chat message. I'll just check once. Thanks. So now we know that there are accessory cells or antigen presenting cells that have pattern recognition receptors. And you can imagine, so these are part of the innate immune system, and the lymphocytes are part of the adaptive immune system. And I'll come to that later on. But again, the infectious non-self model was also not very successful in explaining a lot of clinical contexts like graft rejection, because during graft rejection there is not much infection happening. At least clinicians try to prevent it. And also there are spontaneous abortions in many pregnancies, and you can't really explain infectious non-self. The infectious non-self model can't really explain these clinical contexts, and that led to another path-breaking immunologist, Dr. Polly Metzinger, and who proposed that the immune system doesn't really care about whether it's looking at a foreign entity or it is looking at an infectious pathogen. It only looks at whether there is any danger posed to the body of the host body. And that danger will be sensed by means of some signals which are alarming to the antigen presenting cells, and these alarmins or alarm signals will can be actually responded to by the PRRs themselves. And so she proposed that the pathogen associated molecular patterns can easily be replaced by damage or danger associated molecular pattern. Perhaps the molecules are very similar, but now the only difference is that for danger associated molecular patterns, these molecules are not restricted to foreign antigens only or foreign entities only. There may be host derived alarm signals as well that can drive immune response or activate the antigen presenting cells. And this model is by far the most successful model in explaining a whole lot of clinical context that involves immune activation, and that can explain why a transplant rejection will happen, because during transplant rejection there is surgery going on. Naturally there will be distressed or dead cells, and that will release alarm signals, and that will make the antigen presenting cells license the like foreign antigen specific T cells to react against the transplant, etc. And we also know that in a whole lot of autoimmune diseases, there are host or like endogenous molecules that act as alarm signals or danger signals, which are again sensed by the DRRs or the danger recognition receptors, which was termed pattern recognition receptors by Genway. So there are different clinical contexts where innate immune cells and adaptive immune cells are talking to each other based on whether the innate immune cell has sensed any danger in the host body. And you know about clinical context also in our households, like for example, gout. You can actually, the immune system is actually reacting against monosodium urate crystals, and there is no foreign pathogen involved, but there is a very heavy immune response. Also, there are metaflammation where people are getting fat, and in the visceral fat, there is inflammation happening without any bacterial infection or something. So danger model is by far the most successful model, but I would point to you that here, with the advent of accessory cells by Cunningham and Lafferty, we actually were looking at a cell that was interfacing between the environment and the adaptive immune response. Now, adaptive immune response um, is a property of higher animals, and so if you look at the animal origin or animal classification, you can actually see, so the adaptive immune response, T cells, B cells, come from the jawless fishes, and so then comes the antigen receptor diversity and adaptive immunity, because antigen receptor diversity is the mainstay of adaptive immunity. Because if the immune response has to adapt to antigens that they are looking at, they must be able to either change their antigen receptor based on the antigens they're encountering, and also, or either they have to have a huge diversity of antigen receptor that can actually statistically look at one new antigen and recognize it. Now, there is this watershed moment before jawless fishes, and so what was there before that? Before that, it's mostly the innate immune cells, which in higher organisms we are mentioning as accessory cells. We actually came to know about them from the transplant experiments as accessory cells present in transplants. But these cells are present in lower animals. And for example, in starfish, and which was looked at by Mechnikov, right? So Mechnikov looked at phagocytosis by starfish larvae cells, and so he thought that the phagocytosis is the major work that the immune system cells do. More or less at the same time, on the left is Paul Ehrlich, and he was looking at higher animals and looking at toxins and antitoxins. Essentially, he was looking at antibodies and antigens, and he thought that it's very specific on the and phagocytosis was not something that he was looking at. And why did that happen? Because they were looking at different organisms. And so Mechnikov was looking at starfish, and that did not have any adaptive immune response, while Ehrlich was looking at higher animals, rodents and humans, and naturally he found all these antigen-specific responses in among immune cells. And now there has to be a breach between these two systems in the higher animals, because they do have antigen specific adaptive response, and also the more primitive and innate immune response, which are largely phagocytes and myeloid cells. And there is one cell which largely is responsible for bridging these two responses and driving the information flow from one end to other, and these are called dendritic cells. I'll briefly talk about these cells, how they were discovered. Now, as I already was kind of mentioning sideways, that innate immune response will actually have, it is a more primitive and they will be very rapid response. They are mostly phagocytic cells. They will, if they see any pathogen, they will try to engulf it and digest it. And it's a very non-specific response because they don't have any antigen-specific receptor. They only have certain receptors that can make them sense some kind of danger. And most importantly, the immunological memory is largely not present in innate immune cells. Although in the recent years, we know about trained immune system among the innate immune cells. I'm not going to talk about this in this talk because this is largely a workshop talk, but the more classical sense of immunological memory, where you'll have antigen-specific cells present in the body, roving the body, and whenever they find their cognate antigen, and they will start reacting. And that kind of memory is not present in innate immune cells. On the other hand, adaptive immune system will take some days to start its response because it has to mature, because the initial clone, the number of cells in each clone is very less. They have to expand after they have seen the antigen and got the danger signal or danger signal licensing by the innate immune cells. But their response is very specific to the antigen because they do have antigen specific receptors. And some of the lymphocytes, mostly B cells, they can actually change their response based on the antigen they're looking at. They can actually make their antigen receptor more efficient and more avidly binding to the antigen, which changes over time. And they do have immunological memory, right? And so this question that what is this link between lymphocytes and macrophages were again first asked by Barnett in late 1960s, because macrophages were being worked with by certain immunology laboratories, and there were so many lymphocyte labs all over the world. And he kind of knew that there must be a link between these two, how to that we integrate these two different kinds of immune responses in the higher animals. And I think the first breakthrough came in mid 1960s, where I mean in 1963, Gustav Nossel, again he was a doctor who turned immunologist, and he first published a Nature paper in 1963 where he did a very simple experiment. So he took antigens and labeled them and injected them into the foot pad of rats and looked at the lymph nodes. And so his idea was that if lymphocytes are can sense antigens, so that he will actually find these labeled antigens in the lymphocytes that are present in the lymph. But what he found that these labeled antigens were not in the lymphocytes. They were actually between lymphocytes. And that was something very interesting. And the labeled, because the antigens were labeled, these between the lymphocyte cells were also labeled. And he actually found that there are long cytoplasmic extensions. He thought they are those macrophages and that are pre-existent in the lymphoid follicles. And they were actually having these antigens, not the lymphocytes. And that was the first clue for us that how this antigen will be crossing through or passing through the innate immune cell and give the information flow towards adaptive immune cells. Interestingly, similar uh, cells with long cytoplasmic extensions had been discovered long back in 19th century by Paul Langerhans, although he found it in the skin, and he actually thought he was looking at nerve cells, because nerve cells do look like this, and because nerves have these dendrites, etc. And he didn't know that he was actually looking at immune cells, but later on we know that he was actually looking at skin antigen presenting cells. Now we call them Langerhans cells. But the real breakthrough came by these two gentlemen, Zunwil Kohn and Ralph Steinman. Zunwil Kohn was a professor in Rockefeller, and Ralph Steinman, after finishing his medicine in Harvard, came to Rockefeller to work with Zunwil, and his focus was trying to find these accessory cells that can talk to lymphocytes. And the accessory cells talking to lymphocytes is what is all about bridging innate and adaptive immune system. And so they were successful in Rockefeller to actually isolate from mouse spleen some of these cells which had these long cytoplasmic extensions. They had very primitive technologies available. This was the microscope that Ralph Steinman used to look at the first dendritic cells in mouse spleen. And what they did, they started using techniques that were again discovered in Rockefeller itself in other floors. And because at that time, in the early 1970s, the density gradient centrifugation was discovered, and also very higher techniques of microscopy were discovered. And so they made use of those labs and actually looked at mouse splenocytes and found these cells. And then so they were again present between lymphocytes in lymphoid follicles, and so Steinman started looking at whether they are indeed able to drive lymphocyte activation. That means whether they are really able to license the lymphocytes to respond to an antigen. So what experiment they did is called, nowadays it's rarely used, but it was even 30 years back it was used very frequently. It's called mixed lymphocyte reaction. So you take the accessory cells or antigen presenting cells from one individual, you take lymphocytes from another individual, you mix them, and because they are not matching, so they will react against each other. This is not an antigen specific response, this is mostly a HL activation, but indeed you can actually find out which of the accessory cells are driving this response most vigorously. And that's the experiment that Steinman and his lab did. This is Michel Nussenzweig, who was a first PhD student of Ralph Steinman, and this is Maggie Pack. So what Maggie Pack published in 1980, that these cells which had these dendritic processes and which they could isolate by using carefully choosing density gradient centrifugation and adherence to the plastic or glass plates, that these cells are much more efficient in driving the activation of lymphocytes in these mixed lymphocyte reaction plates compared to mixture of all whole blood, white blood cells. That means they had some additional benefit and additional capacity to drive lymphocyte activation. Interestingly, at that time, Kayo Inaba was a scientist in Japan. She also independently was looking at these cells, and she knew about Steinman's paper, which was published in JexMed, and so this was the original paper in JexMed in 1978 or 1979, and she actually asked Steinman to accept her in his laboratory so that they can work together. So Kayo came over to Steinman's lab, and she actually showed or established that the very low dose of these dendritic cells are needed to achieve similar kind of lymphocyte activation compared to other kinds of accessory cells which are not lymphocytes but present in the blood and are component of white blood cells. And that's how dendritic cells were discovered. So I usually show this slide So this is a slide from Steinman himself. This was also shown in the Nobel lecture by Nussenzweig, because Steinman, perhaps this audience knows, Steinman never knew that he got the Nobel Prize and for discovering dendritic cells, because when the committee was meeting to decide that he will get the prize, he was in his deathbed and he actually passed away before knowing that he got the prize. But the committee did not know that he passed away. That's why he was given the prize. And the Nobel lecture was delivered by Michel Nussenzweig, who was his first PhD student. And so he also showed this slide, and this is the way Steinman used to describe dendritic cells. So dendritic cells are talking to T cells, and so they are working as sentinels in the body. They're looking at different entities that may be antigens and danger signals. They're activating these dendritic cells, and they're going to the lymph node and talking to T cells to drive immune response. Now, if you look at closely, I mean, of this mechanism, so the dendritic cells are developed from hematopoietic stem cells in the bone marrow. They come to the periphery, but they are not mature. So these immature diseases are roaming around the body, all around the body, going to different tissue places and sampling antigens. And whenever they are sampling antigen in response, I mean, in the context of danger signals, they get mature. So if they are not seeing antigens in the absence, if they are seeing antigen in the absence of a danger signal, they're usually not mature and they become tolerogenic. They try to not present that antigen nicely to the T cell. I'll explain why that happens. And so if they're seeing the antigen in the context of danger signals, they're activated or mature. And this maturation largely involves three things. So first thing is they upregulate their MHC class 2 molecules on the surface. The major character of dendritic cells is they, in the steady state itself, they also have very high amount of MHC class 2 molecules, but when they're matured, they further upregulate the MHC class 2 molecule expression on their surface. Second is they start expressing molecules like CD8T, CD86, CD40, which have their cognate receptors on T cells. Now, MHC class 2 molecules will load the processed antigen as peptides on them and will talk to the T cell receptor with T cells. And that's how the T cells will get to know about the antigen recognition or antigen identity, whether that their TCR is good for that antigen or not, good for that epitope or not. But these receptors, I mean, these molecules called CD86, 80, CD40, there are a few more other molecules, B7 molecules. And we have cognate receptors on the T cells that can actually interact when the T cells are meeting with disease in the lymph node. And when they do, the T cells are activated most vehemently. Now, this is the link between danger signal and T cell activation or efficient T cell activation, because only danger signals or DAMs activating pattern recognition receptors on the disease will actually upregulate these molecules, which are co-stimulatory molecules. Now, the third most important effect of maturation on disease is expression of a chemokine receptor called CSIR-VII. Now, CSIR-VII will actually look at chemokines that will give them a chemotactic cue so that they can traverse to the lymph node, to the draining lymph node. From the tissue where they sampled antigen, they go to the lymph node. And so if you put them in the anatomical context, so say immature cells are roaming around all over the body, it may be epidermis, intestine, blood vessels. And if they are looking at antigens in the context of danger signals, they will get mature and they start their journey towards the lymph node. And in the lymph node, they will talk to T cells and drive T cell activation. So you can actually look at this is a live microscopy from Michael Kahalan lab in UC Irvine. So this is a dendritic cell which doesn't have any, which has not seen any antigen. Nevertheless, it had reached the lymph node, but you can actually see it's not interacting with a lot of lymphocytes. They're just it's just roaming around in the lymph node. It will come out and it will come back to the periphery. But when they're loaded with antigen and they're also expressing co-stimulatory molecules, you can actually see there are tight interactions and with a whole lot of lymphocytes coming towards it and actually interacting with it. And that's where lies the importance of co-stimulatory molecule expression on the antigen presenting cells. Now, as I said, the co-stimulatory molecules have cognate receptors on the lymphocytes. Now, some of these co-stimulatory molecules are activatory that I already talked about, and but some of them are also inhibited. That means when they engage the cognate receptor on the lymphocytes, they give a negative signal. And so all of us now know, because of cancer immunotherapy being in clinical use for last decade or so, about PD-1, PD-L1 interaction. So there are other negative co-stimulatory molecules which also can drive a negative signal in the lymphocytes. And that's how these dendritic cells try to prevent autoimmunity, because if they are looking at self-antigen in the context of absence of any danger signal, they will go to the lymph node, but they will actually upregulate these negative co-stimulatory molecules. And while talking to T cells, they will give this information that I'm not really activated in a way that you should be activated to drive inflammation. And that's a major tolerance mechanism, peripheral tolerance mechanism that is working all over the body all the time till we are alive. And so now there are different kinds of different subsets of dendritic cells that have been now discovered, and I'm not going into details of them. I'll show you there are at least one decade was there when every other month a new dendritic cell subset was being discovered. Now we know it's there are some functional subsets, there are some ontogenic subsets. That means they are kind of designed in a way from the ontogeny or hematopoietic stem cell proliferation itself. And so there are major divisions are two kinds of cells. One are called the conventional disease. Here it's a myeloid disease. It's called being called, but this is an old terminology. We call them conventional disease that were discovered by Ralph Steinman, and I was talking about them till now. And there is also another subset which is called plasmacytoid disease, and they are also dendritic cells, and they also play a big role in bridging the innate and adaptive immune response, but they do it in a little different way. They do using whole lot of type 1 interferons or specific cytokines that can actually have its effect on different lymphocytes and other adaptive immune. Now, I'll just briefly talk about the discovery of PDCs. They were almost at the same time. So 1978 was the time when Steinman published his conventional DC paper. I mean, near about that time, Giorgio Trinchieri in Philadelphia, working on cells, he was trying to find out cells in the human peripheral blood that will produce interferon alpha in response to viruses. And he found out some cells which were non-lymphocyte, non-myeloid, non-NK cells. And he also found that they are MHC class 2 positive. And he also found they were not able to present antigen very efficiently. But what they were able to do was producing interferon alpha or other type of interferons, interferon beta, in response to virus infection or even to bacteria. And but he did not know, because the conventional dendritic cells were also being discovered right at that time. So he did not name them dendritic cells, but he actually described the right properties of these cells. But the final description came from this guy, Youngjun Liu, who published a science paper in 1999, long after Trinchieri actually described these cells. And he characterized them as one subset of dendritic cells, and they're most efficient type of interferon producers. They're much less efficient in antigen presentation. And he also found that in a lot of contexts, these cells, these dendritic cells, are rather tolerogenic. they would rather not activate T cells, and they will drive T regulatory cell expansion. And interestingly, these cells have their pattern recognition receptors mostly inside the endosomal compartment. The highest number of molecules of pattern recognition receptors are TLR7 and TLR9 in these cells, and they are all endosomal toll-like receptors, and they usually recognize nucleic acids, and that's how they produce type 1 interferons in response to pathogens. And people have now found that they can also recognize nucleic acids of self-origin, and that's how they can actually drive a whole lot of autoimmune diseases. Again, this slide comes originally from Yongjun Liu. And so, as I said, so at some point of time, there were different subsets of dendritic cells being described in different tissues and different clinical contexts. And mostly they were differentiated based on their surface markers, what are the proteins that are present on their plasma membrane, because those proteins will be used to recognize them on flow cytometry experiments, and also specific transcription factors that were driving these lineages. Now, there are really viable questions whether they are really lineage commitment or functional plasticity. You know, a dendritic cell is moving around the body and in some tissue in response to some tissue factor, it is plastic enough to express certain proteins on its surface, and that's how they take a different surface marker conformation rather than other surface marker conformations. Now, it's not very clear for all the subsets, but some of the subsets are now well characterized to be driven by master regulator transcription factors and expressing stably some of the surface markers on them. And also, there is a very interesting division of labor among these conventional dendritic cells. For example, PDCs I already talked about, the major function is producing type 1 interferons, and type 1 interferons have a major role to play in maturing B cells and antibody producing B cells. There are CYP11B positive cells in mice, which are in human, we call it BDCA1 positive cells, and they have specific transcription factor master regulators, and they are mostly presenting antigen to the CD4 T cells based on MMC class 2 peptide loading. And there are other kinds of cells which are more efficient in cross-presenting the CD8 T cells. And so I don't want this audience to get more into this classification, but I am just trying to impress upon you that there are divisions of labor, right? So for even these innate adaptive bridge mechanisms. Now, if you just take a overall view, so this is your antigen presenting cell, and dendritic cells are the most important antigen presenting cells. I already described them, and they are talking to different lymphocytes, and so they are looking at to the infectious agents and the molecules that are sourced on those infectious agents, or danger signals that are sourced from endogenous cells which are stressed or dying, or they are phagocytosing pathogens and getting their antigens, and then they are presenting this antigen to the lymphocytes, driving effector lymphocytes or helper lymphocytes or B cell activation. Now, I'll add here that there are other established antigen presenting cells which have very important role in specific tissue contexts and specific clinical contexts. In certain inflammatory context in a certain tissue, these cells take up the major antigen presentation roles. But there is a difference. I'll talk about it. So for example, B cells are very efficient antigen presenting cells. They are very important in a whole lot of autoimmune diseases where B cells play a major role in presenting self-antigens. Now, macrophages, monocytes, they have the ability to present antigen, but when they're really optimally activated, when they're really looking at a whole lot of pathogen-associated molecular pattern or danger signals. But for dendritic cells, because they have MHC class 2 expression, even at the steady state of its, I mean, when it's immature, still it's expressing MHC class 2 molecule to a great extent. They have this very interesting ability of driving naive T cell activation. Now, whenever you are talking about naive T cell, you have to know that antigen specific naive T cell is always very rare, right? Because the clonal expansion has not happened. It has not seen the antigen, it has not been activated. Naturally, the frequency of these naive T cells of specific antigen specificity will be very less. And so DCs will have very less opportunity to look at a naive T cell when it is reaching the lymph. But if it has a very high amount of MHC class 2 molecule, if one antigen presenting cell has a very high amount of MHC class 2 molecule on it, even on the steady state, it has a higher chance of encountering naive T cell effectively and driving activation. And that's what makes conventional dendritic cells far apart from the other antigen presenting cells, which can do the job, but only when they're activated and in specific clinical contexts. Now, very recently, there has been ideas about whether this innate adaptive breach can also be mediated by another cell between antigen presenting cells and the adaptive immune cells. And it came from mostly NK cells lab. The idea mostly came from NK cells cell labs, whether they can also breach the innate response with the adaptive immune response, and also NK T cell labs and other kinds of T cells which do not have antigen specific receptors. And now mostly we classify them under one single group, which are, and so lymphoid cells is a very interesting intermediary between the innate and adaptive immune system, and it's a cellular intermediary, so this is not a molecular interaction that is happening. The cellular intermediacy is happening through cytokine production. Now, there are different kinds of innate lymphoid cells. You don't have to get into the details of it, but what is important that they do not express, they are coming from lymphoid progenitors in the hematopoietic origin, but they do not express any antigen receptor which is specific to specific antigen. And neither they undergo clonal selection, like clonal expansion when they're stimulated. And sure. Yeah, I'm almost done. And so they actually, as I said, they develop from the lymphoid progenitors. The same progenitors where from the lymphocytes are being generated, but they directly recruit to the tissues and then they mature in response to cytokines, which are being produced in a specific inflammatory context. And these cytokines can come from stromal cells or epithelial cells that are responding to some pathogen. And these cytokines will drive the innate lymphoid cells to get activated. And when they get activated, what happens? They get activated, they produce further different kinds of cytokines. And these cytokines can actually regulate the activity of dendritic cells as well as adaptive immune cells. And so these cytokines have been shown to be driving different immunocellular effector functions. For example, it can drive macrophage activation, driving ROS generation in macrophages. It can drive epithelial cells in the, for example, in the lung epithelial mucus production from the goblet cells. It can drive extracellular matrix remodeling. It can drive vasodilation, etc. And so here they can actually breach to the adaptive immune cells in two ways. First, they are getting the cues of getting activated from stromal cells and epithelial cells and other accessory cells in the tissues. But when they're activated, they can either drive direct, I mean, there is a possibility of driving direct presentation to the T cells. There are certain studies that have shown that. And also, there are the cytokines that they are releasing that also work on disease, and that indirectly drives adaptive immune activation. So they can actually, these innate lymphoid intermediators can actually drive adaptive bridging, innate adaptive bridging through both direct and indirect ways. And so as I said, so this is a cellular intermediary which is connecting innate immune cells with the adaptive immune cells, largely by producing different cytokines. So I'll mostly end here. So you can actually now see these are the innate immune cells. Some of the cells I did not talk about because of the time. So I talked about the most important one, which is a dendritic cell, which has the innate immune function, and also it actually bridges the innate and adaptive immune system by presenting antigen in the right context of co-stimulatory molecules and specific cytokines that will drive further activation of the adaptive immune cells, and some of the cells will turn into helper cells, some of the cells will turn into cytotoxic T cells, the CD8 T cells, and these helper cells will also drive B cell activation, and B cells will start producing antibodies that are antigen specific. And I'll end here. I'll be happy to answer any question or discuss something more about some of these concepts. Thank you so much.
Yeah, thank you, Dr. Ganguly, for a very lucid talk, bringing in the historic context for the immunology. So we will have time for some questions. Once I hear the question, I will repeat it for you, and we'll go from
Sure.
there.
Thank you.
Yeah.
Thank you so much. I'm so sorry I'm not physically present there.
Yeah. Yes, questions, please.
So the question was about how interferon alpha influences the B cell maturation and activation. How does that work actually? Yeah, and maturation.
So type 1 interferons are of different types. So I was talking about type 1 interferon, which comes from plasmacytoid disease, and there is type 2 interferon, which is interferon gamma. Actually, both of these interferons are able to drive B cell activation. Now, type 1 interferons are very important in first, if interferon is present in the environment, type 1 interferon is present in the environment, the DC mediated T cell activation becomes much more robust. And secondly, where the type 1 interferons also affect the B cells when the B cell is talking to a helper T cell. And when it is talking to a follicular helper T cell, if interferon is around, B cell activation is again much more robust. And finally, when a plasmablast is maturing into plasma cells, at that point also interferon receptor activation drives a very robust response and very robust plasma cell expansion and antibody response. So that's how. So interferon is type 1 interferons are working at DC T cell interface, follicular helper T cell to B cell interface, as well as plasmablast maturation to plasma cells. So in all these ways, it can actually affect adaptive immune activation.
Yeah, please.
Normally, we know that the tolerogenic diseases are usually immature in nature. But as I learned today, even plasmacytoid disease also can induce tolerance. Are they phenotypically very similar or different?
In terms of the outcomes, they are very similar. Phenotypically, they are not similar. I mean, in terms of surface marker expression, etc., they are not very similar. But for example, the major co-stimulatory ligand interaction that drives PDC or like plasmacytoid DC-mediated tolerance in ICOS ligand. On the other hand, for dendritic cell-driven tolerogenicity, it's mostly the CTLA4. And so in that way, so the molecular mechanisms or molecular interactions are a little different, but outcomes are largely similar. But one difference in terms of the outcome is immature disease at times drive the TR1 cells or energetic T cells or IL-10 producing T cells, which are not expressing FOXP3. But PDCs are usually, when they are tolerogenic, usually they drive FOXP3 expressing T regulatory cell expansion. And so again, the PDC driven tolerogenicity is again very much tissue context driven. For example, they have been demonstrated in tumor most often, and also during in oral tolerance, in oral hypersensitivity to allergens, etc., which are orally taken in the oral mucosa. So these are the two contexts where PDC-driven tolerogenicity is most documented. But on the other hand, the conventional DC-mediated tolerogenicity is more ubiquitous. Almost in any tissue context, any antigen context, uh, they can actually work. So they work in tumors, they work in gut mucosa, they work in lung mucosa. There have been seminal work demonstrating the tolerogenic function in different tissue contexts for conventional disease. But PDC-driven tolerogenicity is most important in the cancer context, inside the solid tumor, and people have shown effect of TGF beta, which drives PDCs to a tolerogenic phenotype. People have talked about lactic acid, which is more abundant in the tumor that can drive PDCs to tolerogenic phenotype, things like that. And so the end is the phenotypically they are different. The way they drive their tolerogenicity is different. Outcomes are largely similar, barring that the PDCs are only able to drive FOX50 positive T regulatory T cells, as far as we know till date. Thank you.
Yeah, so if there are no more questions, so let us give Dr. Ganguly a big round of applause for a fascinating talk.
Thank you so much.
Thank you. We'll close the session here.
Thank you for chairing the session. And I also thank Ganguly sir for delivering the last speech of the day. Now I request Dr. Ravikant Mishra sir to felicitate Dr. Devinder Sehgal. Thank you for inspiring us with your speech. With this, we come to the end of the scientific talks for the day. I hope these talks have inspired the young generation to look towards the positive aspects of research and think out of the box for carrying this beautiful scientific tradition ahead, adding to more knowledge and bringing more laurels to the nation.